Listeria monocytogenes is in news nationwide. I have prepared this presentation to share with you what I know about this pathogen and how we may help ourselves to avoid infections with Listeria. It is not easy. I encourage you to spend extra time on each and every slide. Try to understand the message I give here. And for example, the rocket in this picture has a special meaning when I talk about Listeria here. We will explain it later. This is a September 2011 news item. 22 people were diagnosed with Listeria, 4 people died. And in our own backyard, Edinburgh, Texas, in the Rio Grande Valley, was in news with the latest Listeria infections. Unfortunately, 20 to 30% of all clinical diagnoses result in human death. This is a serious issue. There is economic and emotional stress associated with the Listeria infections. This is not something new. The cases of Listeria-infected cantaloupe had been going on for decades. It takes a lot of time, effort, and money to get a cantaloupe to a dining table. People love it. One obvious question comes to my mind is that, is the rough skin of some cultivars a factor in carrying the bacterium from the field to the kitchen? Let us talk about the bacterium itself, Listeria monocytogenes, named after a famous British scientist, Sir Dr. Joseph Lister, and also after the white blood cell monocytes, as first found in rabbit blood in 1926. There are several species of Listeria reported. It is rod-shaped. It travels in miniature rocket speed. It can come with almost all food that we love to eat. This is a very important piece of information. Usually, we have a culture of cooking well and refrigerate to minimize foodborne pathogens. But the biology of this bacterium is different. It can grow at 107 degrees Fahrenheit, and it can grow inside your refrigerator. This makes a bad situation for us. But there is more. Listeria can survive in 10 to 12% salt solution. It can survive a wide pH range, for example 4.1 to 9.6 pH. We are dealing with a super smart guy here. We think salt can dehydrate bacteria, but not in the case of Listeria. So, an important thing to remember is that it can come with almost all ready-to-eat food that we love to eat. The people in any one of this category are more sensitive to Listeria. They are children, the elderly, pregnant ladies, and all people with a poor immune system. So this is also a scary situation. Listeria can make people sick. It can make people very sick. It can kill people. In fact, 20 to 30% of clinically diagnosed people die from Listeria infections. Let us see how they behave in our intestine. If we get 5 bacteria from any one of the food items in the tummy, every hour the bacteria will double its number, and they move around. They have 2 key functions for their survival at the expense of the host. One is the rocket, and another one is their ability to kill our enzyme. It is explained better in the next slide. Our immune system is designed to kill all bad guys, including Listeria. So when a Listeria bacterium enters, our defense force will surround it with a wall containing enzymes to kill the bacterium. But this bug has the ability to produce a counterenzyme that will destroy the wall. Then it uses the host cell to make a rocket tail to move around very fast. Think about a police arrests a guy and locks him up, but the bad guy turns around and takes over the entire police station with ammunition and speed. That is what happens here. There is a movie from the Stanford School of Medicine that explains the movement of Listeria inside the host cell.
The difference is that Pathogens, organisms that cause disease, carry genes to encode for a certain number of proteins, a handful of proteins, that are able to specifically interact with proteins in human host cells and elicit very specific kinds of behaviors from those human host cells that result in the signs and symptoms of disease.
And as further explained in this slide, with this type of mechanism with the bacterium, it is not surprising to see many recalls of food items. So what do we do? To solve this big enemy, your high temperature, your refrigeration, acidic or alkaline pH, or even salt treatment is not sufficient. How about a physical treatment with high pressure? Please take time to read and understand this graph and the note provided here. The authors claim that high pressure treatment damages bacterial cells. My question is it viable for us to use in the kitchen? Moreover, what happens to food items when you apply high pressure? Please take time to read and understand the note here. We are dealing with a small size organism. We can line up more than 50,000 Listeria in 1 inch length. Would high pressure wash in the kitchen help? I doubt it. Can we diagnose it properly and accurately before the victims lose their lives? Please read the complications listed here. The take-home message is that first, we accept that it is a difficult bacterium to destroy. Do whatever humanly possible to avoid Listeria infections. The knowledge about the biology of the bacterium, its potential to kill people, and our limitations to counter this bacterium are all true facts. That lead us to only one thing: an awareness-based food safety culture at home, workplace, and at eating places. Sorry, there is no magic bullet. Let us offer our sincere wishes to the victims of Listeria for a speedy recovery, and our condolences and prayers to the loved ones that lost a friend or a family member. Let us hope for a magic bullet from research and development. I'm Manish Karia. Thank you for listening.